Okay, class. So after this timeline experiments, I would be like to move ahead from here. That is the experiment of Hershey and experiment of Maurice Wilkins and Rosalind Franklin. So as I described earlier, that now the DNA has been now considered as the transferred material, the material of heredity, the material which has the ability to transfer from one generation to next generation. Then it was the Rosalind Franklin and Maurice Wilkins. Earlier I have also told the name in the class. They actually proposed something in the year 1952. So this was the X-ray diffraction study carried by them. So for DNA fibers, so they have proposed some kind of a structure like this, but they were unable to make it at a conclusive point. So utilizing this format of their structure, Watson and Crick later on proposed the DNA's double helical structure. So what happened is that James Watson started analyzing the X-ray diffraction pattern as described by Maurice Wilkins and Rosalind Franklin. So there are lots of evidences they started to collect actually. That is the Chargaff's rule. Chargaff says that purine is equal to pyrimidine and ratio is 1:1. And then in the knowledge of the chemical structure, that is presence of deoxyribose sugar, nitrogenous base, phosphate molecule. So by compiling all these things together, James Watson and Crick in the year 1953 proposed the double helical model of DNA structure, and for which they were awarded the Nobel Prize as well. By moving it further ahead, people have started working on human genome project. And in a nutshell, I can say that till now it has been proposed that human genome is actually 3.3 into 10 to the power 9 base pairs. And if you are talking about the number of genes, it is somewhere around 26 to 32,000. So now the terminologies can be described as DNA is equal to, what I can say, it is a nucleic acid. Then what is gene? Gene is the segment of DNA that actually encode the proteins. So what exactly the genome is? It is the entire nucleic acid component of any organism. And nucleic acid, as I told you, they are the polymer of nucleotides. Nucleotides are the monomer or the building blocks of nucleic acids. So what are proteins? Proteins are, as I told you, in central dogma of life, DNA to RNA and RNA to proteins. So proteins are polypeptides which are made up of individual amino acids linked together. So this is the basic skeleton of amino acid. Then I'll be starting with nucleotides in my next presentation. Thank you.